Hello all. Uh, Welcome to week 4 tutorial session. Good evening to all. Other people will be joining. So, am I clearly audible? So, whoever has joined, Nachiket, Charna, Sarath. So, you all have registered for the examination for this course. You can comment and say and tell your thoughts. But registration is still going on, is it? I think it was okay. If registration is still going on, if you like, okay. So you guys are submitting your assignments on time and all. Yeah, if you register, then submit those assignments. And this tutorial sessions is to guide you, to teach you the whatever you have been like sir has taught in the class, and also for the tutorial sessions also. So what I generally do, once your deadline gets over, I just discuss with those questions in the tutorial whatever was there, so that in the main exam you won't face any problem related to that questions. And also for the upcoming tutorials which there is still deadline left, so for that we are not actually supposed to tell the answer beforehand. But if you have any particular question, so from beforehand you can tell me that I have doubt, so I can make your concept clear around that question but won't be able to tell the answer. So today so far we have discussed week 1, week 2 and week 1 and week 2 assignments. Yes, so week 3 assignments I will be discussing today. And also, so right now if you are planning to register, then submit your assignments on time and read through the lectures, previous lectures also, whatever available in YouTube. So most of the assignments questions have been taught in the class and also are being taught by I am also revising those whatever sir is teaching, so that you will get to learn those things twice and get to revise, okay, along with solving answers. So whatever I have seen is that now you have 2 assignments left, week 4 and week 5. So regarding chromatography, so I'll be showing some previous year's question related to chromatography today at the end of the class. And at the starting of the class, what we'll do, we'll revise some concepts which you guys have done in the previous assignments, because most of the things will be again coming to the final exams, okay. So and that will be also very much related to whatever assignments you are having. So you need to have clear concept surrounding that. And also I'll be starting teaching today regarding chromatography. So related to the presentations sir has given, through that only I'll teach you. If you have any particular doubt, if you have already gone through those lectures, then what you can do, just let me know whatever you have doubt, then I can precisely cover those portions also. So right now so what I will do, I will start with chromatography also week 4 contents, and before that I will be discussing week 3 assignments. If you have any doubt, feel free to let me know. I will get back to you with the answers today or the next class, okay. So I'll be starting sharing my screen. What error occurred? Okay. Yeah, I think my screen is visible to you. mm, if only one person you can tell, just yeah, yes or no, because I don't know why it's, is my screen visible or not presentation, right? Some paper is showing up, I think. uh, where is the link? So is my presentation slide was visible? Can anyone tell? It was presentation slide, right? The like week 4 presentation, or it was some paper, random paper was showing up. mm, Paper, okay. I don't know what they're having problem, they're letting me in test. Okay, let me try something else. Just 1 minute, let me try. mm, so no, no, not right. mm, Window. mm, Just let me know if my presentation slide is visible, because there is some error occurring. Now tell me, can you see week 4 experimental biotechnology, something like this? If yes, then we are good to start. No? Okay. What are you, what, okay. Mm, is it visible? It was not visible now? Okay. Stop sharing. I will join from another account. I'm online. Just be there. 1 minute. What happened? Ma'am, your screen is visible but not the presentation. Okay. Okay. I'm seeing. I don't know what's wrong with the Google Meet link. Okay. I will just leave and okay. I will leave and then join soon. You be there. Or if the meeting itself gets closed, then please try to rejoin again. Okay, because there is some technical glitch is happening. I'm not able to mm, present my entire screen. I don't know what is happening. Just 1 minute. I'm leaving and joining back. What's the problem? Sorry for the technical glitch. I'm not able to understand why it's happening. So what I will do, uh, so every time I think you can see the tutorial classes whatever I am taking, I am uploading in a YouTube channel. So the link is there in the list. And also what you can do for today, if you can cooperate like that, I'll be yeah I'll be uploading my presentation slide in the folder link. So tell me, can you access my presentation slides? So there are, you can see there are tutorial session presentation folder. If you see, like if you try go in the tutorial video in your SWAM portal, I will just show you. Where is the SWAM portal? So there I am uploading, and you will just directly go back to the or what I can do, I will just message the link here where I have uploaded the PPT. Then you can open it, and while I will see, you can see it. And if you face any problem, today only I will be uploading the video in the YouTube channel. You just go and be there. I'll try to fix the technical glitch later. Today I will just try to manage somehow. So yeah, I'll be uploading this to the there, so that you can just download it. Sorry. Lecture 4 I'm uploading here, so that I'll just send you the link and you can access it. It's almost done. So by the time, if you have any doubt regarding the assignments which are online or which are already done, you can just comment below, so that we can discuss. But the presentation, I'm sending you the link, you can go through. And for your better understanding, I'll be uploading the tutorial video, because I am recording my screen. So the presentation whatever I'll go through, all those things will be recorded, so there won't be any problem. But in the Google Meet there is some technical glitch happening, so that it's not letting me share my entire screen. Okay, so I think yeah, I have uploaded the file. What I will do, I will send you the link. My drive. Copy link. So this is the presentation link of today's presentation, whatever I will be discussing. In the comment section I have put. So if you can access, you can just click the link. Then whenever I will be discussing, I'll be keep on telling that in which slide I am, so that you can correlate. But due to some technical glitch, I'm not able to share my entire screen. Oh yeah, okay. It's I think it's sorted. I think you are now able to see my presentation slide. I hope. I hope you are able to see my presentation slide. Okay. Yeah, presentation is on. I think, yeah, yes. So I'll be starting with today's class. Sorry for the technical glitch. So hello everyone. If till now you are not able to see, you can comment below. But I think you are able to see. But still, if not, you can tell me. I think you are able to see. Okay. So today's class I'll be starting. Yeah. So so hello everyone. Good evening all. Today is week 4 tutorial session for experimental biotechnology. I am your teaching assistant Ananya Chatterjee from. I'm a PhD student in IISc Bangalore, and this course's instructor is Professor Vishal Trivedi from IIT Guwahati, and the course duration is July to October 2023. If you want to get certified and want to get registered for the course, you can register. So there are some assignments you need to submit, and the maximum marks will be based on the final examination. So in this tutorial sessions, we basically discuss all those assignments, whichever assignments are being given to you in during these courses. So once the deadline gets over, we discuss the answers, so that you can correlate whatever you have given answers, and what was the problem you can ask me. So you week 1 tutorial session was completed. Week 2, week 3 is completed. Assignments 1 and 2 I have discussed. This is the link for all the YouTube videos, all the tutorial classes, and this is the folder link of all tutorial slides. Whatever I discuss in the class, in this link I upload. So without further delay, I will start with week 3 assignment. So week 3 assignment was live. Is there any problem happened? Any questions so far? Okay. So week 3 assignment was live, and the deadline is over. So I'll be discussing the answers and the solutions with you. So there is a question: a protein shows a molecular weight of 70 kDa in SDS page and 140 kDa in native page. What is the oligomeric status of the protein? So in those scientific question session, we have you have seen that what is the use of SDS page and native page. So by the time you know well, is there any question? Not visible. Okay, I can't able to see slides. Okay, till now it's not visible. How can it be possible? Stop presenting. Minus a PowerPoint. Can you see now? Okay, then I think I know what to do. Just a second, I think I know. Done. Can you see now presentation slide? I think you can. I hope you can. Or okay. Or what you can do? Stop presenting. Chrome tab. Share tab audio. Select a tab to share. Can you see now? Okay, perfect. Okay, let's start. Okay, I hope you were able to see. If not, then please unmute yourself and tell me so that I won't go back and see. If yes, also you can unmute. Someone can unmute and say that yes or no. Starting.
Yes, ma'am. Now it is
Oh,
visible.
okay, sure. Thanks. So that's all. So week 1 tutorial 2 and 3 are completed. Assignment 1 and 2 has been discussed. If you have any doubt, please let me know that these questions you have doubt, and I'll be able to help you in solving and clearing your doubt. So this is the YouTube link which you get in your SWAM portal also. SWAM portal also you get this link for those tutorial sessions. You can go back if you miss class, or if you want to revisit those tutorial classes, you can go through these links and then attend, just listen to the lectures. The folder, all the slides I use in my tutorial, all slides I have been downloading uploading in this folder. Okay, so today we'll start with week 3 assignment, which you have already submitted, most of you. So we'll be starting this discussing. So if my cursor is visible, so the question is, you have run a gel. Okay, so for now you have understood that what is gel, why you run those, why you run proteins and all. So in this question they have told that a protein you have run in HDS page. So basically HDS page, what is that? It has a denaturing tendency because it has HDS. It denatures the protein, it gives a linear negative charge, so that the protein is completely in a linear peptidal form. It's not like secondary structure, tertiary structure. It's not at its native state. So in that HDS page you have got a band of 70 kDa, and you have run a native page. And what is native page? We have discussed in the previous classes. So native page exactly where HDS is not present, where BME is not present. So basically in native page you can see the oligomeric states of your protein. What is oligomeric state? If a protein monomeric is 70 kDa and the protein has a tendency to dimerize or tetramerize, so all those domains will be together, right? But in the case of HDS page, we are breaking those bonds to separate those oligomers into monomers. So only we can see 70 kDa bands in the case of HDS page, because we are denaturing and reducing those domains. But in the case of native page, we are not denaturing, we are not adding HDS, we are not heating. So what will happening? The protein will be intact in its native conformation while running through. For that the molecular weight will be same. So in that case you have seen 70 kDa in HDS page and 140 kDa in native page. What do you think? That it will be what kind of oligomeric status? So it has just doubled. Double means it is what? It will be 2. So you have to calculate that how much multiplication you are getting. If it's thrice, then you would get 21 kDa, right? So if it is oligomeric, it is dimeric. It is becoming dimer. So answer will be 2. Accepted answer is 2. Now the next question is, vertical streaking of protein band in HDS page occurs because of what? So options are overloading of protein sample. So you know, while you load your protein sample, there is space. Okay, it's like there is some spaces. So it can hold up to 15 to 20 microliter based on the comb size. You put your comb and you can like, so you put your comb and you can make those gel wells. So there is space. I am just trying to find if there is a pen or something I can strike through. Okay. So never mind. So what we do, there are combs, so that space only you can able to load your protein. So if you load more than the capacity of the well, what will happen? There will be vertical streaking. What is vertical streaking? you can see that there is a line, vertical line, which is like a streaking appearance. So this features you can see where the protein bands appear due to overloading of the protein sample, and you can correct this can be corrected by either reducing the amount of protein sample or running the gel at lower current. Okay. Then the third question was, appearance of distorted protein bands in SDS page can be troubleshooted by. So if you run gel, you can sometimes see that there are some smiley bands, there are distorted bands, there are vertical streaking, but you want to troubleshoot those. So what are the possible uh, troubleshooting parameters you can apply in this case? So options are decreasing the amount of TEMET and APS, increasing voltage of power supply, increasing amount of TEMET and APS, decreasing the voltage of power supply. So the answer will be you have to increase the amount of TEMET and APS to polymerize your matrix, that is page, polyacrylamide gel robustly. So what happens, appearance of distorted or uneven protein band is due to stacking gel polymerization. It can be corrected by increasing amount of ammonium persulfate and TEMET or de-aerating the stacking gel. So if some aeration happens in that stacking gel, you can do that also. So you have to ensure that there is no like air bubble going on and something like that, so that your proteins are stacking properly. Any doubt in this cases? If not, we'll discuss further. So while you will run gel, you have to do some troubleshoot. You may face that your protein bands are getting smilier. Like smiley means it will be like this. So those proteins which I have load at the left will be up at very up, and the right will be at very up, and the like the middle one will be forming a curve. So that is the thing. You may face smiling, you may face diffuse protein bands, you may face vertical streaking, you may face that proteins are running faster than expected. So what you will do in these cases? For smiling, what happens, uneven heating of the gel causes differential migration of proteins with outer lens moving slower than the middle lens. Rapid heat transfer eliminates this defect and can be achieved by filling the lower tank with a buffer until sample height. Okay. So sometimes what happens, you are running the gel, okay, you are applying some voltage, and this will create heat. So if heat is not properly transferred everywhere, so some portions will be much heated where those electrodes are. So that's why those positions where those electrodes are getting much more heated. So significance of low current. Okay, so significance of low current is your gel will run slowly. Okay, you are applying low current, low voltage, that means your gel will run slowly. So those, like, it will help you in properly resolving. If you run very fast, heat will also be generated, right? If you run very high voltage, high current, heat will be generated, so there will be distorted bands. So that's why lower current, lower voltage will help. So now diffuse protein bands, how it happens? So diffuse pattern of the protein bands appears on the page sometimes. So this diffuse protein bands pattern can be corrected by increasing running current by 25 to 50% higher concentration of acrylamide. So sometimes if you are running at very less current, that is also not good, because you are letting the protein sitting through the gel for longer time. So you have to have the proper voltage and current. It's not like that you will run very high current, it's also not like that you will run at very lower current. So diffuse means what is happening whenever you are loading your protein in the gel. So the gel is a matrix, it is porous, so the protein is also will diffuse by its own way, right? It along with the electrostatic along with the electrodes, like electrical field, it will also try to migrate by itself only. So if you are running very slowly, then the protein diffusion will be increased. but you can't also run at very high voltage also, so you have to have the optimum thing. And also diffuse protein bed, why do you think that higher concentration of acrylamide will help? Because if you increase the concentration of acrylamide, what will happen? The pores of the acrylamide will reduced. Okay, so acrylamide forms like this kind of porous structure, right? So if you increase the concentration, the pore will be less, so the rate of diffusion of proteins will be less. Now the third problem is vertical streaking. Vertical streaking of the protein bands appears due to overloading of the protein sample. Yeah, that's what we are discussing. So it can be corrected by either reducing amount of the protein sample or running the gel at lower current. Fourth, protein runs faster than expected. In few cases, migration of protein is not proportional to the molecular weight. It is either more or less on gel than the expected place. It's due to the unusual very high proportion of basic or acidic amino acids. So suppose your protein is of 50 kDa, but you are not seeing it's exactly at 50 kDa. You can see it's 55 kDa or something like that. So what is happening? It's either more or less on the gel than the expected place. So it can be due to very high proportion of basic or acidic amino acids. Okay. And also sometimes if your protein is glycosylated, some proteins are glycosylated, post-translational modification happens, due to those glycosylation also molecular weight increased and those proteins are uplifted. Okay. So yeah, now we'll move next. Double bands. Appearance of double bands is due to partial oxidation or degradation of the protein sample. Addition of more amount of reducing agent or preparing a fresh sample will reduce the artifacts. Okay, so sometimes also you can see double bands in very same size, maybe very if your protein is of 60 kDa, you can see like 60 and 62 kDa, you are getting a double band. So this can be also possible that due to partial oxidation is happening. So when you are making the sample, your sample buffer has BME. So BME, over the time BME gets oxidized. So you have to make fresh sample buffer so that this reduces these artifacts. Second is distorted protein bands. Appearance of distorted or uneven protein band is due to stacking gel polymerization. It can be corrected by increasing amount of ammonium persulfate and TEMET or aerating the stacking gel. So the same thing. So you have to add APS and TEMET so that you are not your stacking gel polymerization have been happened successfully. Now the next parameter is lateral spreading. You can see that your proteins band are getting laterally spread. So it can be avoided by reducing the time between loading of the sample and running. So it's again the diffusion problem. So what happens if you are loading the sample and you are running very late, you are running after 1 or 2, or what will happen? It will start spreading laterally. So yeah, it will start spreading laterally. Now we'll move on to the next question, which is unfolding of a protein can cause us dash, which makes its mobility slower in the SDS page. So what can be the answer? Option 1 is increase in charge. So if a protein is getting unfolded, can it be possible that it's increasing in charge? No. Increase in molecular mass, this is also not possible. And folded protein, unfolded protein, both the molecular mass should be same. Increase in hydrodynamic volume and decrease in hydrodynamic volume. So what could be the reason? If a protein is folded and if it's folded small and getting unfolded, so the hydrodynamic volume, what that means, the protein surrounded by water will be increased only, right? Because unfolding is taking more space. So the answer will be increase in hydrodynamic volume. Next question. Dash atoms are used for leveling DNA for visualization in autoradiographs. So for DNA, for leveling DNA, what you can use? You can use radioactive active isotope of phosphate, because DNA has phosphate in its backbone, right? The next question is, which of the following technique is used for purification of radioactive labeled oligonucleotides? So you can do gel filtration chromatography, we will discuss in chromatography classes. So the answer is gel filtration chromatography with Sephadex G50 column. So regarding the isotope labeling, approximately 30 picomole of each oligonucleotide is dissolved in sterile distill buffer for further labeling. So you label with alpha 32 phosphate labeled ATP, because for DNA has ATP and GTP and like that. So if you want to label ATP, what you will do, you will use alpha 32P labeled ATP. Okay. Yeah. So the next question is, the next answer is, the labeled oligonucleotides are purified by gel filtration on a Sephadex G50 column. The column is pre-equilibrated by passing 10 ml of 10 millimolar tris pH 8 through it. Okay. So this is the answer. You have to while you are purifying the labeled oligonucleotides, you have to do gel filtration on a Sephadex G50 column. Okay. Now we will move towards the next question. Animate all. Yeah. So the next question is, what is the role of T4PNK in gel filtration? Sorry, in gel mobility shift assay. So PNK is a kinase. So what is the function of kinase protein? It helps in phosphorylation. Okay. So the answer will be, it catalyzes the binding of radioactive ATP to DNA oligomer. Okay. So it is doing the kinase activity, showing the kinase activity. Now you have heard about depth treated water. What is depth treated water? Depth treated water is this is very pure water you use to inactivate RNAs. So while you work with RNA isolation and work with any RNA experiments, you have to be very careful, because RNAs is present everywhere. It can be present in your hands, it can be present in your desk. So it's present in everywhere. So you want to make sure that you are degrading all the RNAs, enzymes, and you are saving your RNA. So you add depth-treated water to inactivate RNAs. Next question is, in super shift experiments, which is added to bind the protein prior to forming DNA protein complex? So what is added in super shift experiments? So what happens for the super shift experiments, for the protein, certain amount of antibody against the protein is also added. So what is shift experiment? To briefly tell you, so that you know what is gel mobility shift assay or what is super shift assay. So what happens when we run the gel? Okay, we load DNA or we load protein, but suppose some protein is binding with DNA and you want to see whether DNA protein complex is getting formed. So what you will do, you will run only DNA once for control, only protein for control, and you will run DNA plus protein complex for the experiment. So what you will see, if the protein is binding with the DNA, it will reduce the migration rate of the DNA, because the molecular weight has become larger in this case. So it will reduce the rate, so that protein will run a bit slower than those negative control proteins, negative control protein and DNA lengths. Okay. And for super shift experiments, what kind of protein, particularly what protein is getting bound, that also you will be able to know, because here, like Western blot, you are adding respective antibody against that protein. Okay. So in super shift experiment, what you will do, for this experiment, 2 microgram of respective antibody is added to the binding reaction mixture prior to the addition of label probe. This mixture was incubated for 20 minutes. Then the label probe is added and incubation carried out for another 45 minutes. So like that, you add your antibody to the reaction mixture prior to the addition of level probe, and you do this experiment so that you get to know the protein also, specific protein. So the answer will be 2 microgram protein specific DNA, sorry, antibody. So the next question is, what percent of gel is used for analyzing the samples in gel mobility shift assay? So in this case 5% non-denaturing polyacrylamide gel is used, so you need to have the pore size a bit higher, so use 5% non-denaturing polyacrylamide gel. So why non-denaturing? I know you can able to tell now, because you don't want to denature if there is some bonding happening between DNA and protein, right? Now the next question, what was that? Which of the following extracts would you use to identify a transcription factor for a gene X? So uh, suppose you were giving an interview and people are asking that you want to isolate one transcription factor. So what do you think, from which compartment of cells you will get? Definitely nucleus, right? Because transcription and all, whatever you will be getting from a gene, so it will be in the nuclear extract. Okay, because the transcription happening there, so it's pretty much clear. Now the next question is, DNA's footprinting is performed to. Now what is DNA's footprinting? DNA's footprinting is that we want to know at which position of DNA some protein is bound. Footprinting means the protein has given its footprint. Okay, so DNA's footprinting means, what do? We identify the exact protein binding sites on DNA. So answer is C. So how do we do that? We treat with DNA so that the DNA gets cut out, but in the space where protein is getting bound, DNA can't access that space. So what happens, that DNA becomes a bit longer. So from that we can identify that this is the size where the protein DNA could not be chopped off, where protein was bound. So yes, so this is the, yeah, for those T4 PNK gel mobility shift assay, these are the reagent and materials you want to use. Like you have to use 5% non-denaturing gel, you have to use 32 phosphate labeling, reaction mixture, 5x binding buffer, and rest of the things are same. So the next slide is, question is, what is used to denature secondary structure in RNA? So secondary structure in the RNA is formaldehyde is used. Okay, formaldehyde is used for denaturing the secondary structures in RNA. Which staining dye is used in RNA agarose gel? So DNA and RNA, everything you can stain through ETBL. So answer will be ETBL. The next question is, which is used as a de-staining solution for RNA agarose gel? So if you are staining with ethylium bromide, what do you think you can use as de-staining solution? So what you use is that 0.5 molar ammonium acetate. So you have to know this. Now, so this is another assignment for chromatography previous years. So we will be discussing that. Before that, I will be discussing chromatography concepts, so that you will be able to answer those. So this is the slide from Professor Vishal Trivedi, who is taking the courses. So what is there? Let's discuss. Chromatography. So what is chromatography? The first true chromatography is okay. Do you have any question? You can just write down in the comment section. So how do KD values affect illusion order? Illusion or is decreasing or increasing KD values? Okay, so you have to understand what is KD value is. So for that we'll discuss the chromatography principle. So you can see what is KD value. So a distribution of a molecule between two phases A and B. What is the use of different types of chromatography? What is it? Okay, hold on. So this is a formula for KD value. A distribution of a molecule between two phases A and B is given by a distribution coefficient. So KD value is basically distribution coefficient. KD equals to concentration in phase A divided by concentration in phase B. So in most of the column chromatography technique, phase A is stationary phase or matrix and phase B is mobile phase or buffer. Okay. So I'll be discussing in details, don't worry. And what do you mean by different kind of chromatography? What another person has told? What is the use of different kinds of chromatography? So yeah, I'll be discussing. So there are several kinds of chromatography based on what do you want to purify. Okay, so you will apply that method. So basically the first true chromatography is usually attributed to the Russian Italian botanist Michael Savit. So literally means color writing. So chromatography means color writing. We used to do in those is KD physical or chemical property, on what factors does it depend? Okay. So what do you think? What is KD? I will come to this. So what do you think? So you in the chromatography what happens? Two phases are there. One is matrix, which is stationary, and one is your buffer, which is moving. So your molecule, the distribution of molecule where it is being present in phase A or phase B, that is the principle of KD, the distribution coefficient. Okay. So in example you can see that how, so this is the sample mixture you are injecting, and in time to time where it is getting present in which fractions. So yeah, I think now we'll be able to answer the questions. Okay, so yes, so why we are using different kinds of chromatography? Because we want to we are applying several techniques to isolate to purify particular things. So chromatography is a separation technique to isolate the molecule of interest, and that depends that, so there different kinds of chromatography comes into play. Now the thing is that there are mixture of molecules. There are mixture of molecules and pure molecule you want to separate. So what is the meaning of this separation or isolation? You want to understand. Okay, so that is why the chromatography is. So principle of separation you have to discuss. So you will be able to answer that is physical property or chemical property, because we'll be discussing the separability, the solubility, the KD one. So what, now understand this slide first, that this is three molecules. Okay, now how two molecules can be separated from each other? These three molecules, which is benzene, phenol and aniline, are similar to each other but have distinct physical and chemical properties which can be used as a criteria to separate them. So what do you do in column chromatography? We try to separate our molecule of interest in the basis of their physical and chemical properties. So the physical and chemical properties which can be used to separate are physical. What is physical properties? Is molecular weight. Note down please, what are the physical properties? It's molecular weights, boiling point, freezing point, crystallization, solubility and density. From this you can tell that KD is what? Physical property or chemical property. Okay. What are chemical properties? Functional groups. For example, phenol has OH group, hydroxyl group it has, whereas aniline has NH2 group. Number 2, reactivity towards other reagent to form complex. If it's reacting, then it's chemical properties. If it's solubilizing a different thing, that is physical property. Okay. Now, principle of separation, you can have that its molecular formula this and this, molar mass are different, densities are different, melting points are different, boiling points are different, but these boiling points are little bit similar. So you have to analyze this physical and chemical properties so that you get to know that where the difference is and you can purify likewise. So here what they have done, they have mixed benzene and aniline at begin, and then they did condensation. So they have isolated the benzene and aniline is there, and at the end they have done the aniline and benzene separately. So you can see the difference. Okay, so they have boiling point is different, benzene boiling point is different, aniline boiling point is different. Okay, if you are heating the sample at 100 degree, the aniline will not boil, but benzene will boil, and then condensation will happen. Okay, so the distribution coefficient to describe the distribution of compound 1 between phase A and phase B is what you were asking, Narayana, is Kd equals to concentration is phase B divided by concentration in for phase A divided by concentration in phase B. Now, the purpose of chromatography is to separate a complex mixture into individual components, exploiting the partition effect which distribute the molecules into the different phases. Different forms of chromatography there are, which are partition chromatography. In this form of chromatography, an analyte distribute themselves into two phases, liquid, stationary and mobile phase. Okay, so there are two phases. One is stationary phase is liquid, and so there are two phases, liquid, stationary and mobile phase. So it can be stationary and mobile. So there are two phases. The major advantage of this chromatography is that it is simple, low cost and has broad specificity. It is further divided into liquid-liquid chromatography because both the phases are liquid. Bonded phase chromatography, example, gas liquid chromatography, thin layer chromatography and paper chromatography. So these are the partition chromatography. So it's liquid, gas liquid, thin layer and paper chromatography. Adsorption chromatography. In this form of chromatography, matrix molecule has ability to hold the analyte on their surface through a mutual interaction due to different types of forces such as hydrogen bonding, electrostatic interaction, van der Waals. So these are, so where adsorption means something is getting bound. So while in the chromatogram column you are applying something, and then there are some stationary phase which is interacting with your sample, that means they are absorbing the sample. So there is some interaction going on, that's hydrogen bonding, electrostatic interaction, van der Waals. So these examples are ion exchange chromatography. So this example, ion exchange chromatography, hydrophobic interaction chromatography, affinity chromatography and so on. So in this case, interaction will be happening based on their properties with your sample. So this kind of chromatography is adsorption chromatography. Now next, a distribution of a molecule between two phases, while we have discussed Kd equals to concentration, how much concentration of that molecule is in one phase and divided by is in another phase. So now coming to the mechanism of separation in chromatography. So what is column chromatography? So in this chromatography, a stationary phase is filled into a cylindrical tube made up of glass or steel. The mixture of analyte is loaded into the top and runs from top to bottom. So there is a tube which is which has a stationary phase filled, okay, and it can be a glass tube or steel tube. So now the sample mixture which you want to separate, this yellow and red you want to separate. So the sample mixture you load into it, you inject it. So So yeah, so for protein, if you are dealing with two phases, it depends on where the affinity of the protein is. Okay, if you are working with some sample and working with different density, you so so your molecule where the density is matching, your molecule will go to that. So that is how KD is there. Okay, and also if so the separation thing na, for protein you have asked. So what is the thing is that you have used two phases. So in one phase they have a tendency to bind to your protein, but in another phase they don't have. Then you will calculate likewise. Now in this example, assume there are two molecules X and Y with a KD value of 1 and 9, and they are traveling through a column with water as mobile phase, and as they will travel, X and Y will partition between stationary phase and mobile phase. As there is a huge difference in KD, Y will be associated with the matrix and remain on the top on the column, where X will move along the water. At the end of the chromatography, X will come first. If you understand this example, you won't have any doubt regarding KD. So what is happening? Say you have started working with two molecules X and Y, and KD is difference. There is huge difference, and one is 1 and another one is 9. So that means one molecule has much affinity towards the solution phase and one has much affinity towards the stationary phase. So likewise you will be able to isolate, because what will happen that Y, in this case Y will be associated with the matrix, Y will be associated with the stationary phase, and it will take time to come out from that. But X has KD value is 1, so it will come out faster. Okay, so this is the concept. Now from chromatography you get chromatogram, the plot of elution volume along with the absorption. Yeah, you have asked that how from the KD value you can determine elution order. So this is the answer, that so you know the KD value. What happens? The KD value was less in the case of X molecule, it has come out early. Okay, the elution has happened earlier. So if you know the formula, you will be able to interpret. So here what is happening? This is a baseline. You have injected your sample. This is detector signal. So this is detector signal. It can sense your the amount of protein you have and on the other basis. And this is time. Within the time, what molecules is getting coming out earlier. So the plot of elution volume along with the absorbance is known as chromatogram. The volume or time it takes for analyte to come out of the from the column is known as retention volume or time. So the volume or time it takes, so how much time it takes is called the retention time, and how much volume it takes for analyte to come out is called retention volume. So here you have seen molecule A, B, C, D has come out. So A has come out at the very beginning, then B, and then C and D. Now coming to the resolution. Resolution means how well you are able to separate two molecules. So the ability of a chromatographic column to separate two analyte peak from another is known as resolution. It is defined as a ratio of difference in retention time between two peaks, an average of base of peak width. It's given, so there is a formula of resolution. This is delta retention time divided by WAV. So where is defined as a ratio in difference in the retention time between two peaks, an average of base peak width. So this WAV is the average peak width. Okay, so when a resolution is 1, the separation of two peaks is 97.7%, and a column with RS more than 1.5 considered good. The number of distribution events govern the ability of a column to separate the two analytes. In other words, resolution is directly proportional to the number of distribution events. In column chromatography, each thin plane of column matrix participate in distribution of molecule. Assume height of a distribution plane is h and length of the column is L. Hence, number of distribution in plane is n equals to L by h. If it's getting complicated, no need to worry. It's not you don't need to think in a complicated way. It's just like you need to know Okay, so what is the effect of urea while adding to protein? I have already discussed in the last class. Urea helps in unfolding protein or denaturing protein. So what do you do in urea gel? You add urea and you see how much amount of urea you are adding, so that you are seeing that this molar, 1 molar or 2 molar or 6 molar or 8 molar urea is required to completely denature your protein. Okay, I hope your answer is you can able to understand. So this is the effect of urea. So now so this is the retention volume. You need to understand what is the resolution, that's all. This is nothing much. So there are two formulas in this is not so much complicated. That if you there is height of H and length of the column is L, then N will be the distribution plane, that there are how much plane is there, that's all. Resolution now comes that hence the number of distribution plane in a column is controlling two parameters. One, as number of distribution plane will go up, it will allow the analyte to travel for longer period of time. Consecutively, it will increase the distance between two peaks. 2. As number of distribution plane will go up, it will reduce the width of the base of peak. As a result, the peaks will be more sharp. A representative example how number of distribution plane affects the base of the peak is given. As the number is increasing, the peak width is decreasing. Hence, the number of distribution in an indirect way to measure the column efficiency higher in number is desirable for better separation. It's just like if your volume of the column chromatograph is higher, you are able to separate more. So relative absorbance here, n is 1000, n is 100, n is 10. So it depends on the how much you are resolving. You can see that the resolution for n is to 10 is not good, right? Yeah, resolution of n is to 10 is not good. You can see a broader peak. For n is to 1000, it's a longer peak, like that. It's a resolution. So this is a chromatography system setup, where at first you have one reservoir. So you can have one or two reservoir for mobile phase, based on how many mobile phases you are adding. Number 2, you have pump, one or two pump to flow the buffer from reservoir. Different types of pumps are used in chromatography system, mostly based on the pressure level required to perform chromatography. Mixture. A mixture is required to mix the buffer received from both pumps to form a linear or step gradient. So this is a mixture. So where you can mix both the buffer. Fourth, sample injection loop. So you inject the sample here. Fourth is column, a column made up of glass or steel. So there you inject your sample and buffers and everything passes from there. Then after that, those samples whichever is getting eluted, it will go through the detector, where the elution coming out from the column goes to the online monitoring system to test the presence of the analyte based on different properties. There are different types of detectors are known in chromatography, such as UV visible detector, etc. So in the detector that will detect your data and analyze, and your fraction will be collected. So fraction collection, the element can be collected in different fraction by a fraction collector. And recorder, the profile of eluent with respect to the measured property in a detector can be plotted in the recorder. So this is a ACTA pure chromatography system. It's basically those simplest picture. Here is your reservoir. Here you can attach your column. There is detector. Here is mixture, pump, and then those are fraction collector. Now what you can do, if you have any doubt, you can let me know. What happens when we add okay, unfolded protein elute first or last? I mean treated or untreated protein in chromatographs. So what do you think? If you are unfolding your protein, the protein is become linearized, and if you are folding your protein, the protein is in a globular form. It's taking lesser hydrodynamic radius. So if you are increasing your size and hydrodynamic radius, it depends on how it's getting eluted. So you have to think it. If the question is directly in assignment, I won't be able to tell you the answer directly, but you have to think. So in the SGS page, I can tell you what happens in the SGS page. If you run unfolded protein and then folded protein, what happens? What comes first? And also you have to understand that if your protein is in oligomeric condition and you are adding urea, that will denature. So that will be in monomeric, it is reducing. So if molecular weight is getting decreased, you can tell based on that whether it will come first or last. It's based on which kind of chromatography you are performing. Okay, if you are performing size exclusion chromatography, then what will happen, that small molecular weight will come last, like that. So you have to think like that. Okay. How is absorbance happens? What happens when we add ligand to protein? If we add ligand to protein, the protein will, if there is protein has affinity towards ligand, it will bind to the ligand, and then the size will be increased. Then you can be able to tell if size is getting increased, what will happen, right? Okay. Now we'll be discussing about ion exchange chromatography. Yeah. So this is a protein. It can be purified through affinity, it can be purified through charge, through surface area, through hydrophobicity. So different chromatography techniques. One is ion exchange chromatography, another one is hydrophobic interaction chromatography, gel filtration chromatography based on surface area, and another one is affinity chromatography. Okay, so you can purify your protein based on 4 factors: charge, hydrophobicity, surface area, and affinity. Now ion exchange chromatography. So the principle is the chromatography distributes the analyte molecule as per charge and their affinity towards the oppositely charged matrix. The analytes bound to the matrix are exchanged with a competitive counter ion to elute. The interaction between matrix and analyte is determined by net charge, ionic strength, and pH of the buffer. Okay. So when a mixture of positively charged analyte M plus, M minus 2 loaded onto a positively charged matrix, the neutral or positively charged analyte will not bind to the matrix, whereas the negatively charged analyte will bind as their relative charge and needed higher concentration of counter ion to elute from matrix. Okay, so if you have a matrix which is positively charged, and if you have ions mixed of positive charge and negative charge, what will happen? Those negatively charged ion will be binding with the positively charged. Okay, so types. No, it doesn't remains in column. It depends on what kind of chromatography you are doing. It doesn't remains in column. It will come out, right? The linear protein. It will elute. So there are two types, cation exchange chromatography and anion exchange chromatography. So the matrix used in ion exchange chromatography is present in the ionized form, will reversibly bound into a bind ion to the matrix. The ion present on matrix participate in the reversible exchange process with analyte. Hence, there are two types of ion exchange chromatography. Cation exchange chromatography. In cation exchange chromatography, matrix has a negatively charged functional group with an affinity towards positively charged molecules. Okay, so here you have negatively charged functional group and has affinity towards positive charge. So the positively charged analyte replaces the reversible bound cation and binds to the matrix. In the presence of strong cation in the mobile phase, the matrix bound positively charged analyte is replaced with the illusion or illusion of analyte. Okay. Anion exchange chromatography. In anion exchange, matrix has a positively charged functional group with the affinity towards negatively charged molecules. The negatively charged analyte replaces the reversible bound anion and binds to the matrix. What is reversible bound? That is, the anion is getting bound with the matrix, but the reaction is reversible. It can also unbound, get unbound. Okay. In the presence of a strong anion such as Cl- in the mobile phase, the matrix bound negatively charged analyte is replaced with the illusion of analyte. Now, list of selected ion exchange matrix. Type strong cation, ion exchange group sulfonic acid, pH range 4 to 30. Column matrix you can use sp cephros, sp cephedex, cm cellulose cephros. Weak cation which has carboxylic acid 6 to 10 pH. So strong anion, weak anion. Strong anion you can use quaternary amine, weak anion primary amine, secondary amine, tertiary amine, DEAE. So quaternary amine or strong anion example is Q cephros, capto, ambertyl. For weak anion, DEA cephros, capto DEA, DEA cellulose. So these are for weak anion. Ion exchange chromatography. So this is cation exchange chromatography. Matrix where is negative. So if positive proteins are getting bound, what happens? You can elute by controlling the amount of Na plus. Okay. Anion exchange chromatography, what is happening? Positively charged matrix. If negatively charged protein is bound, you can elute with the amount of with the help of amount of Cl minus. Operation of ion exchange chromatography. Selection, you have to select what kind of matrix sorry, what kind of matrix you want to use. You have to prepare the matrix beads for chromatography. Operation of IEF chromatography. So you need to calculate the pi value of the protein and net charge, structural stability, enzymatic activity. Selecting matrix, pi value and net charge. The information of a pi will allow you to calculate the net charge at a particular pH on a protein. So you can calculate how to you know how to calculate the pi of a protein. So a cation exchange chromatography can be used below the pi sorry, whereas an anion exchange chromatography can be used above the pi value. Okay. So if a pi is there, so cation exchange chromatography you can use below the pi. Okay, and anion exchange chromatography you can use above the pi. So you can calculate your pi, calculate the pi of your protein in XPASI website. So you have to just give the sequence of your protein amino acids, and then they will calculate the pi for you. Experimental calculations. Proteins have minimum solubility in solutions with pH that corresponds to their pi and often precipitate out of solution. Okay. So so there are several pH, that is 3, 4, 5 and to 10. So proteins what happens in the pi range, what happens that proteins charge is neutral. So it has minimum solubility in solutions with pH that correspond to pi, and that's why they get precipitated. Selecting matrix in IEF. So you want to consider pi value and net charge, structural stability, enzymatic activity. So the information of a pi will allow you to calculate the net charge at a particular pH on a protein. A cation exchange chromatography can use the below pi and anion is above pi. Structural stability. 3D structure of a protein is maintained by electrostatic and van der Waals interaction between charged amino acid, pi-pi interaction between hydrophobic side chains of amino acids. As a result, protein structure is stable in a narrow range around this pi, and a large deviation from it may affect its 3D structure. If you are not, so you have to have a narrow range surrounding your pi for the structural stability. Enzymatic activity. Similar to structural stability, enzymes are active in a narrow range of pH, and in this range should be considered for choosing and ion exchange. Similar to structural stability, enzymes are active in a narrow range of pH, so in this range should be chosen for your enzymatic activity. Preparation of matrix beds for chromatography. Swelling of medium. Swelling makes the functional groups to be exposed for ion exchange. The swelling of anion exchanger is usually carried out by treating it first with an acid 0.5 normal HCl and then with base 0.5 normal NaOH. Exactly the reverse is the case with cationic exchangers. The matrix can be treated with EDTA for impurity eliminations. Removal of very small particles. These fines will decrease the flow rate and the unsatisfactory reaction. To remove fines, the exchanger is repeatedly suspended in a large volume of water, and after the larger polymers have settled down, the slow sedimenting material is decanted. Equilibration with counterions. This is accomplished by washing the exchanger with different reagents depending upon the desired counterion to be introduced. NaOH counterion to be introduced is sodium, and HCl you want to add counterion to be introduced HCl. So this is a matrix. These are fixed ionic groups on the matrix. So these are the buffers for cation exchange chromatography. The citric acid, lactic acid in this pK value, and working pH are also given. And these are some buffers for anion exchange chromatography with the pK value and the working pH. So column material and stationary phase. Column material should be chemically inert to avoid destruction of biological sample. It should allow free flow of liquid with minimum clogging. It should be capable of withstand the back pressure, and it should not compress or expand during the operation. Positively or neutral charged protein. This is negatively charged protein, this is positively charged protein. So mobile phase, the ionic strength and the pH are the crucial parameters to influence the property of the mobile phase. So sample preparation. The sample is prepared in the mobile phase, and it should be free of suspended particle to avoid clogging of the column. The most recommended method to apply the sample is to inject the sample with a syringe. There are many ways to elute analyte from an ion exchange column: stepwise gradient, continuous gradient, either by salt or pH. Increasing the ionic strength using a gradient displaces bound proteins as ions in the buffer compete for binding sites. Elution further increases in ionic strength, displace proteins that are more highly charged. So column regeneration after the elution of analyte, ion exchange chromatography column require a regeneration step to use next time. Column is washed with a salt solution with the ionic strength of 2 molar to remove all non-specifically bound analytes and also to make all functional group in ionized form to bind fresh analytes. So final high ionic strength wash removes any ionically bound proteins before re-equilibration. So application of IEF chromatography: hardness removal, dealkalization, heavy metal removal, fluoride removal. So yeah, application of IEF. So there is application in the case of softening water. So groundwater has several metals such as calcium plus 2, magnesium plus 2 and other cationic metals. So a cation exchanger matrix with bound sodium is packed in the column and the hard water containing calcium magnesium is passed through the column. So what happens in this process? Calcium present in the solution preferentially migrate from the solution to the matrix, whereas sodium ion present in the matrix migrate to the solution. The matrix can be used for softening of the water until it has bound sodium ions. Once sodium ions are exhausted, matrix can be regenerated by flowing a solution of sodium chloride or sodium hydroxide. The calcium magnesium bar to the matrix comes out. Okay. So ion exchange chromatography purification of proteins from plant sources. So you have to purify the proteins. Stationary phase DEE Cephatics A50 anion exchange chromatography, eluent with the NaCl gradient in 0.1 molar phosphate buffer, detection 280 nanometer, protein bands ranging from 94 to 10 kDa molecular mass, water extract of N sativa was prepared, dried and powdered. Powder was dissolved in PBS insoluble pellet valley. This is how you purify a protein from plant source, and then you use for a chromatography. Used in chromatography. So this is how to purify a protein from animal source. So from fresh egg, they are purifying protein like that, and they are doing chromatography. So this is a research problem. Scientists have discovered a transcription factor in skin cells binds to a set of gene X, which is responsible for changing its color due to exposure to sunlight. Now they would like to identify the transcription factor from skin cells. So what they have to do? I have to isolate the transcription factor with so the transcription factor will bind to the promoter. So this protein binds to the DNA for modulating gene expression. So here DNA is a negatively charged. So what we'll do, this is the experimental design. So protein DNA complex is forming. So in this approach, anion exchange matrix is incubated with the DNA and allowed it to bind tightly. Now the pure protein is passed through the DNA bound bits, followed by washing with a buffer to remove unbounded proteins. Now the DNA is eluted from the matrix either by adding high salt concentration or with denaturing condition. Now the fractions are tested for the presence of DNA and protein. Eluted protein is analyzed in the SDS page and DNA naggled. So in an anion exchange, you are letting your DNA to bind, then you wash with elution salt. So you are using salt to elute your DNA along with protein, because these proteins are getting bound with the DNA. Okay. So there is a second research problem. Cancer cells are treated with curcumin and it activates several protein kinases in cell lysate. Now a PhD student wants to measure total kinase activity of cell lysate. So this is substrate with ATP. If you add protein kinase, then this substrate will be phosphorylated. Okay. Now what you have to do in experimental design, it's become phosphorylated. In the protein kinase assay, a radioactive substrate, preferably a radioactivity on carbon, was incubated with the enzyme protein kinase, magnesium chloride and non-radioactive ATP. A negative control is also been included where enzyme protein kinase is absent from the assay mixture. Reaction mixture from negative control and experimental will be loaded on two separate cation exchange chromatography columns to bind unphosphorylated substrate from the reaction mixture, where as phosphorylated radioactive substrate is present in the flow through. The radioactive count of the flow through was measured using a liquid scintillation reagent. Okay, so what is happening? Those from the control and as well as from the experimental design, you will be loading those substances in the column. So will be loaded on two separate cation exchange chromatography column to bind unphosphorylated substrate from the reaction mixture, whereas phosphorylated radioactive substrate is present in the flow through. Okay, so here the phosphorylated phosphate are coming in the flow through, and all the non-radioactive and the without phosphorylation, those are getting bound with the column. Okay. So this is the unphosphorylated one is getting bound with the column. Now hydrophobic interaction chromatography. So different chromatography techniques, ion exchange, hydrophobic jellyfish and affinity. So we'll be discussing hydrophobic interaction chromatography. So hydrophobic interaction chromatography exploits the ability of a strong interaction between hydrophobic group attached to the matrix and hydrophobic patches present on an analyte such as protein. Okay, so there can be some patches of hydrophobic regions in the protein. So it exploits the ability of a strong interaction between hydrophobic group attached to the matrix. So this, if there is hydrophobic patches, it can be able to bind to the hydrophobic group attached to the matrix. So this is hydrate shell. This is the solubility curve, salt concentration. This is salting in and salting out. This principle will be explaining. So here you are adding salt, and when you are adding more salt, so this is the salting out preparation. So it's getting isolated. So what happens? Addition of low amount of salt when you are starting adding the salt in low concentration to the protein solution results in the displacement of bonded water molecule with an increase in protein solubility. This effect is called salting in. In the presence of more amount of salt, what happens? Water molecules shielding protein side chains are displaced completely with an exposure of hydrophobic patches on protein surface to induce protein precipitation or decrease in protein solubility. This effect is called salting out. So there in the presence of more amount of salt, what is happening? The water molecule is shielding the protein side chains, and so that those are not interacting with the hydrophobic patches. The phenomenon of salting out is modulated so that the addition of salt induces exposure of hydrophobic patches on protein but does not cause precipitation or aggregation. The exposure of hydrophobic patches facilitates the binding of protein to the nonpolar ligand attached to the matrix. When the concentration of salt is decreased, the exposed hydrophobic patches on protein reduces the affinity towards matrix, and as a result it gets eluted. Okay, so the exposure of hydrophobic patches on protein exposure of hydrophobic patches, what it does, it facilitates the binding of protein to the non-polar ligand attached to the matrix. So if your matrix is of hydrophobic, then it will help in binding. When the concentration of salt is decreased, when you are decreasing the salt concentration, the exposed hydrophobic patches on protein reduces. So that's why the affinity towards the matrix gets reduced, and the result is protein gets eluted. So the choice of HIC matrix table, selected list of popular HIC column matrix. So column material: butyl S-saffrose, phenyl saffrose, captophenyl saffrose, octyl saffrose. Functional group: butyl, phenyl, low density phenyl, high density phenyl, octyl. Choosing a suitable HIC matrix is essential to achieve best result. The strength of the binding of analyte on a HIC column is governed by the length of the aliphatic linear ligand. Matrix with a aromatic ring containing ligand makes additional pi-pi interaction, and they will bind analyte more strongly than some same number of carbon aliphatic ligand. In addition, presence of pi-pi interaction gives selectivity as well, such as ring containing aromatic ligand phenylalanine. At last, ligand density plays a vital role in the strength of binding of an analyte to the matrix. Hence, this point should be considered to choose suitable matrix for purification. So this is the HIC matrix. You have some proteins which has hydrophobic patches also. So while it is in high salt, what is happening? This hydrophobic interaction is happening, and while it's low salt, the interaction is not happening. So what you have to do? You have to equilibrate the column material packed in the column and equilibrate in a buffer containing 0.5 to 1.5 molar ammonium sulfate. The salt must be below the concentration where it has salting out effect. Sample preparation. The sample is prepared in the mobile phase and it should be free of suspended particle to avoid clogging of the column. The most recommended method to apply the sample is to inject the sample with a syringe. Elution. There are many ways to elute an analyte from the hydrophobic interaction column: decreasing salt concentration, changing the polarity of the mobile phase such as alcohol by a detergent to displace the bound protein. Column regeneration. After the elution of analyte, HIC column require a regeneration step to use next time. Column is used washed with 6 molar urea or guanidine hydrochloride to remove all non-specifically bound protein. The column is then equilibrated with mobile phase to regenerate the column. The column can be stored at 4 degree in the presence of 20% ethanol containing 0.05% sodium azide. So this way you can store the column for its regenerate, for its good property and quality. So so this was it for today's discussion. If you have any specific doubt, you can ask. So as I was telling okay, so the person who was asking question, Hari Narayana, is there? I think he's not there, so I'll be discussing some previous year question answer that might help you in solving ongoing assignment. So the question is, the partition coefficient Kd depends on what? Physical properties of the molecule, chemical properties of the molecule, both physical and chemical properties, neither physical or chemical properties. So the answer will be both physical and chemical properties, because we discussed right now that there is certain properties, like some are physical, some are chemical. Okay, so the Kd depends on which chromatography you are using. So in a general way, it depends on both physical and chemical properties. Second question. Salting in is a process in which salt increases the solubility of protein. Salt exposes hydrophobic group of proteins. Salt induces protein precipitation. Salt decreases the solubility of the protein. So salt, in the process of salting in, salt increases the solubility of protein, okay? But in the process of salting out, salt decreases the solubility of protein. The interaction, that's why I have not discussed this beforehand, because if you get to know the concepts, you will be able to answer properly. The interaction of proteins with HIC column involves what? Ionic interaction, covalent interaction, hydrogen bonding, hydrophobic interaction. HIC column means hydrophobic interaction column chromatography, so that means hydrophobic interaction. Okay, the The use of high salt concentration for binding of proteins into an HIC column is based on the principle that high salt concentration helps protein in forming hydrophobic hydrogen bonding with the column matrix, removes small molecular weight proteins that interfere with binding, removes hydration layer of the proteins and exposes their hydrophobic residues, precipitates the proteins which stick into the column matrix. So the use of high salt concentration removes the hydration layer of the proteins and exposes their hydrophobic residues, which helps in binding. Elution of bound proteins from HIC can be achieved by decreasing the salt concentration. So that means that is salting. Sodium azide is used in column chromatography as it helps in prevent microbial growth while storing the column. The chromatographic technique is used to separate analytes based on the size of gel filtration chromatography. So whatever chromatography is based on the size, that is gel filtration chromatography. The KD values of the following proteins are given below. Protein A equals to 2, protein X equals to 6, protein Z is equals to 9, protein Y equals to 4. If the mixture of the proteins will be loaded on the chromatography column, the protein dilute at last will be protein Z. Which of the following is a type of adsorption chromatography? The answer is affinity chromatography. The increase in height of the distribution lane will result in what? The increase in height of distribution plane H will result in decrease in resolution of the column. Which of the following technique is best suited for purifying native protein? So that is blue saffrose chromatography. The chromatography technique employs separation of molecule based on electrostatic interaction, that is ion exchange chromatography. Purification of a tagged protein X expressed in E. coli can be achieved in single step, which is affinity chromatography. So if the protein is tagged, it has affinity towards a certain matrix. In an anion exchange chromatography, the matrix is opposite, positively charged. Okay. In hydrophobic interaction chromatography, the salt concentration of the equilibration buffer should be above the salting out effect. Specific interaction between substrate and enzyme occurs. The hydrophobic patches on the analyte interact with hydrophobic groups of the matrix. This is the correct answer. Okay. So this is part 2. We have not covered totally, but we can go through. So the physical property of a molecule utilized in gel filtration chromatography is surface area. A denatured protein has lower retention time as compared to its native state due to increase in hydrodynamic surface area. Increase in gel filtration column length would result in better resolution of peaks. So all these things we have discussed in today's class. A nuclear protein is involved in regulation of gene X. The protein was incubated with gene X and run in a gel filtration column and the chromatogram recorded. Select the correct statement. Protein ligand complex would have lesser retention time. The KD value of the protein ligand complex will be more. The peak corresponding to the protein ligand complex will show right shift. The hydrodynamic surface of the protein will be lesser. A nuclear protein is involved in regulation of gene X. The protein was incubated with gene X and ran in a gel filtration column and the chromatogram recorded. Select the correct statement from the following. Yeah, there is a problem in the question. They don't have any question only. So we need to check. The question has some problem. The nuclear protein is involved. That is fine. And this protein is interacting with gene X. Okay. So here it's written that gene X is the ligand. Okay, gene X is a ligand. So if you are running in the gel filtration column, protein ligand complex would have lesser. No, yeah, there is some problem in the question. They have not stated properly. 3 proteins A, B and C have molecular weight 180 kDa, 50 kDa, 250 kDa respectively. The protein with lowest KD value is protein A, protein C or protein B. So the highest molecular weight is of protein C. So the lowest Kd value will be of protein C. If a protein is interacting strongly with the stationary phase of a gel filtration column, uh, then what will happen? The elution volume will be more than total column volume, because it's interacting very strongly, so you have to add more amount of liquid so that it comes out. Protein purification strategies utilizes physical properties of a molecule except viscosity. Yeah, it depends on affinity, charge, size and shape. Elution of molecules from gel filtration chromatography column occurs in decreasing order of molecular size. Okay, the Kd values of 4 different proteins are given below. Protein S 0, 1, 1.5, 0.5. The mixture of these proteins is loaded on a gel filtration chromatography. Which protein will come out first? So protein A will come out first because it has lowest Kd value. Got it? You can relate to this question also, because in the gel filtration chromatography the larger molecular weight come out first, so that's why it has lowest Kd value. Now, yeah, the molecular weight of a protein was found to be around 120 kDa by gel filtration chromatography. The purified fraction on SGS page analysis result in a band of 40 kDa. That means oligomeric state how much? 40 into 3, that is trimer. If the length of a gel filtration column is reduced by half, the Kd of a particular analyte would remain unchanged because there is no role of the length of the column in the case of Kd value. Which of the following show a linear relationship within gel filtration chromatography? Logarithm of molecular weight and Kd. A protein X from MTB is giving a single peak in Cephedex S200 with a retention volume of 12.63 ml. The protein is treated with an increasing concentration of urea, and the retention volume will be less than 12.63 ml with increasing urea concentration. A protein X treated with 8 molar urea would most likely elute after native protein. Elute at the void volume, will not elute but get absorbed. Elute at twice. Elute at the void volume. The proper packing and presence of bubbles within the gel filtration column can be detected by incubating the column at 40 degrees Celsius for 30 minutes, resolving an analyte with Kd equals to 1, washing the column with organic solvents, resolving the column in a gradient ammonium sulfate. The proper packing and presence of bubbles within the gel can be detected by if you resolve an analyte with Kd equals to 1. Okay, then you will be able to understand the proper packing and presence of bubbles. So the part 2, regarding the part 2 we have not studied yet. So today I have covered the week 4 tutorial session, which is chromatography part 1. Part 2 I'll be covering the next day, so that you will be able to better understand those answers of the question. If you have some doubt regarding that, the elution urea thing he was asking from the assignment, so yeah, that was the answer. Okay, so if you have further any question, you can just let me know, else we'll stop today's class. Thank you for cooperating. We had a technical glitch at the starting of the class, but yeah, thanks for understanding. We'll again meet next week. We'll discuss chromatography. Okay, Narayana, you were here, so I have explained your doubts actually. Whatever you had that illusion, when the illusion will happen, and after adding urea, so it will elute at void volume. And what you were asking, please explain ligand binding to protein. Ligand binding to protein means okay. What graph you are telling? This graph or there is no graph in my slide. What graph in gel filtration chromatography? Okay. Oh, gel filtration chromatography is in week 5 class. Narayana, we have not started gel filtration chromatography yet. I have till hydrophobic interaction chromatography we have studied. So we'll be discussing in the next class. When is your assignment deadline? Can you please tell? We'll be discussing in the next class gel filtration chromatography. If you have any specific question related to any specific question, that you can tell, we can discuss, because I don't have any graph in my PPT. We'll be discussing next week. Do you have any question from here? Okay. Which of the following show a linear relationship within gel filtration chromatography? Okay, so is it elution volume or and kd? Is it elution volume and kd, elution time and kd? Is it logarithm of elution volume and kd, or whether it's logarithm of molecular weight and kd? So in gel filtration chromatography, what happens? The larger the molecular weight, it elutes faster. Okay, so if so larger the molecular weight means the kd value, it's elutes faster, that means the kd value is less. So the elution volume, so the linear relationship between which has a linear relationship, it's logarithm of molecular weight and kd is a answer for this question. So if you want to see the graph, I think through I also need explanation for urea in gel filtration chromatography. Okay, so this thing is that gel filtration chromatography, just hold on, because I have not it's not in my slide. I have not prepared the slide for, because this is for next week. Just hold on a second if you want clarification today. It is in week 5, so just hold on. Gel filtration. Are all chromatographic techniques different? What is their role compared to other techniques? What if you understand from the chromatographic techniques? You have understood that with respect to charge, shape and size, hydrophobicity, we are separating those molecules to separate compounds, right? So what do you mean by what is the role compared to other techniques? Other techniques means what do you want to so if you want to purify, so you will be doing chromatographic techniques because your molecule or compound will have affinity towards certain matrix. Experiments in biotechnology. Okay, so what specific experiment you can do to specify like to purify your proteins or any macromolecules? I will just get back to you with the gel filtration chromatography where it is. Gel filtration chromatography. Actually, we are supposed to discuss the gel filtration chromatography on the next class. Let me see. So regarding gel filtration chromatography, we'll be discussing in details next week. But when is your assignment deadline you want to look for? You want to purify, so if you want to purify some compounds, so that's why you do column chromatography. This is the application. Okay, so Okay, please mail your doubts and questions, because you can also in the discussion forum also you can write, because the chromatography thing, the second gel filtration chromatography will be discussing in the next class, because it's in week 5. We are now organizing week 4 session. So for that I don't have slides for gel filtration chromatography. I will get back to you. You can email in the discussion forum. Yes, sure. Whatever questions you have doubt, look, the questions directly I will guide you with the approach, but directly the answer we won't be able to tell you. So yes, whatever you have doubt, just mail us. Just mail us. Will so that and mail us beforehand, so that we'll. Okay, you are not able to find in lecture notes. Um. Okay. Also, what you can do from the assignments, whatever you have doubt, no, just one day before, uh, you can mail me. I can give you my mail address, and also the discussion forum you can mail. So in this email address you can mail, so that I will be able to know that which portion you have doubt. So then I can discuss that beforehand, because we are not supposed to directly tell the answer. So I am not covering whichever assignments are. Can you rely on other sources? Will they match answer? Yeah, it will match. It will match answers. Yeah. And if something happens, so sometimes some answers you are not confident on NPTEL portals that this answer may not be correct. So what will happen? Your marks will not be deducted. You will be given marks if there is some anomaly in questions, because there were some questions they had anomaly. I think I will look into it twice and will get back to you. And other sources here, you can Google, you can go through some other books, some other videos. So these are general and standard techniques. Yes. You were in bachelor's or master's? In which standard you were? Okay. So if you are doing some practical dissertation or something, you will be able to do in your hand. So if you do these experiments, no, you will uh, understand better. In experiment, do the differ. So is it like the process it's very straightforward, it looks like, but while you do the experiments you will be able to understand better, because you have to do troubleshoot and all. It doesn't differ. It's general standard, but some amount like buffers and all, this all you have to calculate according to your own macromolecule. So what kind of pH you will use, that all depend on your whatever macromolecule you are working on. So it's not that it will work for everything. It's that much standard is not like that. It's you have to optimize for your own experiment. Yeah. Okay guys, thanks for joining. We'll be again next week. We'll be meeting with gel filtration chromatography along with all the assignments, chemical concentration, techniques, formulas. Yeah, formulas are standard, techniques is standard, but chemical concentration is not. You have to optimize it. If 1 molar NaCl is not suitable for your protein, you have to increase it. So like that. But it's pretty much standard. Yeah, the principle is standard. So that's all for today. Thanks for joining. Next class Tuesday 6 to 8 p.m. we'll again meet, and we'll discuss about your assignments, which are not live. Actually, the live assignments we won't be able to discuss. But if you have any principle doubt in principles, I'll be happy to discuss. Um, so yeah. And what else? Yeah, gel filtration chromatography, part 2, which is week 5, which is also live. I'll be discussing that. Okay, thanks for joining. See you. Bye bye. Sometimes, what do you mean by sometimes they differ? So all these techniques are same. Okay, the principle of chromatography is same, but the how much NaCl, how much your particular buffer you will be adding, that you have to understand your, yeah, depending on what experiments you are doing, what do you want to do, that's all. But it's not like that you are working with different macromolecules and all will be same. It's not like that. So the principle is same, but based on the principle you have to design the experiment, that's all. Okay, we'll discuss again next day. Thanks for joining. I'm signing off.